mTOR, the mechanistic target of rapamycin-tyine, is arguably the most important single protein in muscle biology. It's the master regulator of protein synthesis, the convergence point for virtually every anabolic signal the cell receives, and one of the most studied proteins in exercise physiology and cancer biology. Understanding how creatine activates mTOR and how that activation compares to other anabolic stimuli clarifies both the power and the limits of creatine as a muscle builder. mTOR exists in two functional complexes, mTOR C1 and mTOR C2. For muscle growth, mTOR C1 is the relevant one. When activated, mTOR C1 phosphorylates downstream targets, particularly P70S6 kinase and 4E-BP1, that increase the rate of translation of messenger RNA into protein. More translation means more protein being assembled, which means potential for more muscle tissue. mTOR-C1 is activated by multiple upstream signals, growth factors like IGF-1 and insulin, amino acid availability, particularly leucine. Mechanical tension from muscle loading and cellular energy status is sensed through the AMPK-mTOR axis. Creatine activates mTOR through the cell volumization pathway. When creatine draws water into muscle cells via osmotic pressure, the resulting membrane stretch and changes in intracellular ion concentrations are detected by volume-sensitive mechanisms. These mechanisms feed into the mTOR pathway through several intermediates, including changes in the AKT signaling pathway and modulation of AMPK activity. The cell interpreting its own swelling as a positive metabolic signal is an ancient and conserved cellular response. In the evolutionary context, a well-hydrated cell that has absorbed water from its surroundings is a cell in an environment of nutritional abundance, a signal that now is a good time to grow. The magnitude of mTOR activation from creatine-induced cell volumization is real but modest compared to the acute mTOR activation from a heavy resistance training session combined with adequate post-workout protein. Creatine's mTOR signal is not a shortcut to muscle growth without training. It's an additive signal that, when combined with the training signal and adequate protein availability, produces a higher net mTOR activation state than training alone. Several studies have directly measured mTOR pathway activation using muscle biopsy samples stained for phosphorylated P70S6 kinase. They consistently show greater mTOR activation in creatine-supplemented individuals after resistance exercise compared to unsupplemented controls doing the same workout. This is the mechanistic evidence behind the consistent finding in long-term studies that creatine plus training produces more muscle than training alone. Understanding mTOR also explains why creatine without training produces very limited muscle growth effects. The cell volumization signal activates mTOR to some degree, but without the mechanical tension and muscle damage signals from actual resistance exercise, mTOR activation is modest and transient. Creatine is not a muscle-building substitute for training. It's a mechanism that, when running in parallel with training-driven mTOR activation, amplifies the total anabolic signal beyond what either input alone can produce. The full picture. Creatine activates mTOR through volume sensing. Training activates mTOR through mechanical tension and damage signaling. Protein activates mTOR through leucine and amino acid sensing, and insulin activates mTOR through growth factor signaling. All four of these operating simultaneously is the most potent physiological muscle-building environment your body can achieve through nutrition and training alone. The mTOR pathway also has an important regulatory counterpart, AMPK, the adenosine monophosphate activated protein kinase. AMPK is activated when cellular energy status drops, specifically when the AMP to ATP ratio rises, indicating that more ATP is being consumed than produced. When AMPK is active, it suppresses mTOR signaling, shutting down protein synthesis to conserve energy for more urgent metabolic functions. This is the molecular basis for why training to complete failure in a depleted energy state. Very long sessions, inadequate nutrition, extreme caloric deficit can limit the anabolic response despite providing a substantial mechanical stimulus. The cell is too energy stressed to synthesize new protein. Creatine's maintenance of higher ATP levels during exercise through the phosphocreatine buffer reduces the AMP to ATP ratio during the session, keeping AMPK activation lower than it would be without supplementation. Lower AMPK means less mTOR suppression during the session. Less mTOR suppression means the protein synthesis machinery remains more available to respond to the training stimulus.